Okay, so it's my absolute pleasure to introduce the 9th Q-Mare seminar presenter, Doctora Catalina Pimiento. Catalina's academic trajectory is really nothing short of stellar. So originally from Colombia, she conducted her PhD at the University of Florida in 2015, after which she took 3 prestigious postdoctoral positions in Switzerland, in Berlin, and then Swansea. And by 2020, she was a tenured SNF Prima Professor at the Paleontological Institute and Museum at the University of Zurich in Switzerland. And that's a position that she still holds. Now, over these years, Catalina has also worked closely with the institution I work at, the Smithsonian Tropical Research Institute in Panama. And I remember meeting Catalina, what, 12 years ago was that, Catalina? And she had this crazy idea about finding a megalodon nursery. And I have to say, it's an idea which has stood the test of time. And that's really great to see. And I'm delighted actually now to say that she holds a position of research associate at the Smithsonian Tropical Research Institute. So we're effectively colleagues, Catalina, which is great. More than that, she's going to soon hold the position of senior lecturer in biology at the University of Swansea. That's quite an accolade when you look at it. And when you look at the body of the work that she's produced, it's clearly, hugely deserved. So it's a delight to have you join us, Catalina. And today you are going to tell us about the ecological consequences of extinction. It's really an honor. Thank you so much for giving us your time, Catalina. I'll hand it over to you. I think you can just share your screen now. So go ahead.
Thank you, Aaron, for that lovely introduction. I'm going to now share my screen. Again, sharing a whiteboard. We don't want that. Okay, perfect. So hopefully everybody can see my screen. And yeah, thank you again, Aaron, for inviting me to give this talk today. I'm delighted to be here and to see so many familiar names amongst the attendees. So yeah, I'm going to be talking about the ecological consequences of extinctions. And there is a banana plant here, but I'm not in Colombia, I'm in Wales, but I do bring a bit of home to my house. So that's why I decided to sit next to my banana plant. Let me move this a bit here. So we are living, as you all know, in a unique time in life's history. That is because even though life has existed on Earth for 4,000 million years, there have been 5 mass extinctions. Our civilization has existed only for 6,000 years. Industrialization for 200 years only. We're still causing a 6th mass extinction, the first to be caused by a single species, and in such a short time frame. So understanding extinctions is critical. The IUCN, the global authority on ranking species according to their extinction level, states that more than 20,000 species are threatened with extinction. A few years ago, there was a report saying that this translates in almost 1 million or over 1 million species going extinct. So, of course, understanding extinctions is a pressing issue, is critical. What are the most fundamental questions in extinction research? In my opinion, there are 2. One is what makes certain species more prone to extinction. We know that extinction is non-random. Some species are more prone to go extinct than others. And the answer to this question is twofold. In the one hand, certain intrinsic traits can make a species more prone to extinction. For example, for certain clades and under certain extinction periods, body size is a factor that can be associated with extinction, with big species or large species having higher chances of going extinct. Geographic range is also highly associated with extinctions, with species with a very restricted geographic range having higher chances of going extinct. Diets, specialist diets, are associated with extinction risk, especially today. And thermoregulation is also another characteristic, another intrinsic trait that can make some species more prone to extinction, for example, homeothermic relative to ectothermic species. There's also intrinsic factors, external factors, not in the species, but environmental ones that can be related to extinction. So we have temperature causing the extinction of insects, especially bees today. Crashes in productivity have been associated with extinctions, sea level changes, and of course we have human pressures, which we have today. The second question, in my opinion, the most important question for understanding extinctions would be beyond species numbers, what happens when species go extinct, or the ecological consequences of extinctions. So research in ecology, not paleoecology, but ecology that we have today, the animals that we have today, have proposed that there is this thing, this concept of this theory of the world being green thanks to apex predators. So when these apex predators go extinct, their prey, herbivores, overgraze on the environment, and then there's no longer this vegetation, and therefore the world is green thanks to these apex predators that maintain the population of the herbivores on check, and therefore their extinction had these cascading effects across the food web. In the marine realm, we also have similar examples with the keystone species, the sea otter, having once removed either by direct pressure or indirect pressure, and then having an overpopulation of sea urchins, and then affecting negatively kelp forests. So once when some species go extinct, then other species also can go extinct. So this ecological consequences in terms of the cascading effects of extinctions is also very important to understand the ecological consequences of extinctions. Even though we know this kind of the answers or the broad answers to these two questions, our understanding is of course still very limited. In the one hand, even though we know some intrinsic and extrinsic factors can be associated with extinction, until very recently, our understanding on these factors and their association with extinction have been limited to the study of invertebrates, which makes plenty of sense, right? Invertebrates, because a lot of them are benthic, they have shells, they're preserved very well, it's easy to understand them or to study them. However, what happens is that our vision of the intrinsic and extrinsic factors associated with extinction has been focused on this area of the ocean, and we're still yet to see the whole picture of what happens or what are the factors associated with extinction. In terms of the ecological consequences, even though we have this knowledge about cascading effects and so on, a lot of this knowledge is based on extirpations or local extinctions, and they're limited in their geographic range and also in their time range, because of course we can only study what we can observe today with this decline of species. So even though we have a very decent understanding on what causes extinctions and what are the ecological causes of extinctions, our understanding is still limited. I think sharks are great to overcome these limitations for many reasons. I'm going to move my screen a bit here. So first of all, they are very large and diverse. We have over 1,000 species globally, and they're widely distributed around the world. Many of them are also apex predators, and they can control the food webs from the top down, which this is an example here that I'm showing from the Atlantic. So their ecological roles allow us to study the ecological consequences of extinctions. Similarly, and most importantly in my case, modern sharks have an abundant and long fossil record. So a lot of sharks have fossil record that is rooted deep in geological time, makes them a great model to understand extinctions. In fact, Barnowski once said that a possible technique to address the current extinction crisis could be by using taxa best known in both the fossil and modern records. And this fossil record is also very abundant. And finally, or sadly, sharks or elasmobranchs in general are the most threatened marine vertebrate in the world. And unfortunately, current efforts have not been enough, because every year more shark species or elasmobranch species are added as critically endangered or threatened by the IUCN, and we continue to see the decline elasmobranchs in the oceans. But we're going to focus, because I'm a paleobiologist, we're going to focus on the third reason, which is that mother sharks have an abundant and long fossil record. And when I first met Aaron and I was in STRI in Panama, I was doing a lot of field work in the outcrops of the Panama, around the Panama Canal and other areas. And one of the things that really strike me is that even at that time, I didn't have any background in paleontology. I had only studied extant species of sharks. I was swimming with whale sharks in Mexico, and I never thought at that time that I was going to be working in paleontology, and here I am. But at the time, I guess the most surprising thing for me coming from this background was that I was able to identify a lot of the species that we found as fossils, even though at the time I didn't have any paleontological background. And that is because a lot of the species there, this is a picture of an outcrop that is around 10 million years, the Gatun Formation, are still around today. So for example, here, if you can see my mouse, this is a hammerhead shark. This is a lemon shark, this one here. This is a tiger shark. So at least in terms of the tooth morphology, they are the same species as today. And that totally blew my mind. And that is the reason I am a paleontologist today, because I thought that there was such a great potential to understand extinctions by studying the fossil record of sharks. And actually, a recent study that we did recently with a former master's student here at Swansea, we showed that all of the orders of shark species that we have today have a fossil record. Most families, roughly half of the genera that we have today have a fossil record, and around 10% of the species that are extant today have a fossil record. And 10% is of course a low number, but because we have over 1,000 species of shark species, this translates into like 100 and something species having a fossil record. In fact, this graph here is showing the oldest fossil record of extant species. And in terms of species, we have some species that can go back as far as 60 million years ago. And in terms of genera, way deeper. Interestingly, species that are threatened today have a longer oldest fossil record than the ones that are not threatened. And I don't want to give out too much information about this because this is still in prep, but my incredible PhD student, Christina, has been not only, we did this based on the oldest fossil record, but she is looking into the entire fossil record of all species of Neoselechians. And as you can see here, confirming the first study, we show that when we do very sophisticated analysis of all the occurrences, we also find out that the duration of these species that we have today in time zero is very long. So yeah, sharks are super long-lived, and some of them still around today. But I couldn't not talk about the megalodon, the biggest shark ever, which is an extinct species. So not one species that is alive today, that is or extant today, or a clade that is extant today, but one that is long extinct. Because by studying an extinct species, you can also obviously understand a lot about extinction. So of course, I was fascinated by the megalodon when I was in Panama, finding these teeth in many sites that Aaron had been collecting teeth for a long time. And the very cool thing about this species is that, of course, is extinct. And even though it was really, really large, and you could have thought that such a big predator had everything in favor to not go extinct, it did go extinct. And I think that it provides this very good example to understand the consequences of extinctions, because if we translate all the knowledge that we have from recent ecology, and we think that the effects of the extinction of apex predators can have catastrophic effects across the food web, then the consequences of the extinction of these species should have been really important. But the first thing that I did, like Aaron said, was to look at the nursery in Panama. And I had skipped this slide initially, but then I decided to show it anyway, because of course I needed to talk about the nursery in Panama. And yeah, so one of the coolest things ever in the work that I did in Panama was to find or to realize that a lot of these teeth from the Gatun Formation were small, and they were not small because of some sampling issue, but because they were mostly juveniles. It wasn't because they were from the back of the jaw or anything like that. And the Gatun Formation had higher number of juveniles, and that summed to the fact that it was a shallow productive area, allow us to propose a hypothesis that this was a nursery. But that had nothing to do with extinctions, but I needed to talk about it nevertheless. But I really wanted to understand Megalodon's extinction to be able to see what happened when we lost one of the biggest predators of all time. So when, how, and why the Megalodon became extinct. So the first one, when Chris Clemens and I did this work where we used a mathematical model based on the last occurrences to work out when was the most likely time of extinction. And these results suggest that it was around 3 million years ago. We also, with Megan Bolk at the time, both Chris and Megan, we were all grad students. We tested the hypothesis of an increase in body size, because back in the 90s, there was an hypothesis that Megalodon may have gone extinct because it just was growing and growing and growing in size through time, and it just became too big. So we tested this hypothesis, and we showed that actually the body size of the species didn't change through time, stayed the same. Stasis was what we found instead. Then later on, with a much bigger group of collaborators, including my supervisor, Bruce McFadden, and Carlos Jaramillo, we reconstructed the whole fossil record of the Megalodon using all the references that we'll find in the literature, PBDB. Of course, we entered all these references to PBDB and reconstructed all the geographical distribution of the species from the first fossils to the last. And we found, well, before I show what we found, we also did a species distribution model, also to test another hypothesis around back then that suggested that the ocean temperature became too cold, making the environment unsuitable for Megalodon because it liked warm waters. And we found that this was not the case. In fact, the distribution of the Megalodon was not related to temperature. And we found suitable areas for the species even beyond the extinction. So temperature did not correlate or did not drive the extinction of the species. Instead, once we quantified all this geographical distribution, we found that the decline of the species coincided with the decline of potential prey, mammals. We don't really have a lot of direct evidence of what animals were Megalodon prey upon, but it's only logical to assume they were marine mammals, and we do have some evidence suggested that indeed they were marine mammals. So, of course, we have this classic example of marine mammals declining. Marks and Hewen showed these results in their science paper in 2010, declining around the same time as the Megalodon's geographic distribution, and also occupancy or abundance declined around the time. And also the appearance of new competitors coincides with the decline of the species. So instead of being a climatic driven, at least directly, extinction, we proposed that it was more like a biotic factors, so biological factors being correlated with extinction and the decline of the species. And more recently, my PhD student, Jack Cooper, published this paper in Science Advances where we used a vertebral column that had been actually long forgotten in a museum in Belgium. And we 3D scanned it and built the first 3D model of the species based not only on this vertebral column, but also a dentition from the US and a full scan of the full body of a gray white shark to add the flesh, all the components that do not fossilize around the species. And we use a method that has been used before in T-Rex and in other dinosaurs, and we created this. But beyond having this geometric model, and which is cool by itself, we use this, and we use these calculations that we made based on this model to calculate or to estimate better important ecological traits, for example, swimming speed. And we found that Megalodon was able to swim much faster than any other species alive today. But I'm talking about cruising speed, so moving from point A to point B in our case, if we were to extrapolate that to us humans, it would be walking this, walking speed. Nothing to do with burst speed or, the kind of speed you use to hunt. But we interpreted these results as Megalodon having a really great ability to move long distances, migrate, which makes sense, of course, and with that, transport nutrients across oceans. So the extinction of these apex predators, likely had very catastrophic effects in ecosystems, not only because of the top-down control, but also because of the interruption of these migrations. But of course, in order to understand extinctions, you need to look beyond a single species. And that's what I did during my first postdoc in Zurich, where, to be honest, the question behind this other part of my research was if Megalodon went extinct alone or if it was part of a global event. It turned out to be a whole study about marine megafauna, but initially it was all about the Megalodon because I was quite obsessed about it. So yeah, we had reasons to believe that Megalodon didn't go extinct alone. That's because there were many examples of other big animals going extinct around that time. And of course, I also mentioned this study by Marks and Hewen, shows the decline of cetaceans, but also we have other examples of marine megafauna going extinct at that time. So to answer this question, I started a lifelong, I hope, collaboration with Daniele Silvestro, who developed this software Pyrate that uses fossil occurrences, and estimates simultaneously extinction rates, preservation rates, and sampling biases, accounting for sampling biases. And so we use the paleobiology database to download all the occurrences of the marine megafauna from the Cenozoic. In so doing, we also added more references, and we collaborated with people that had been adding occurrences to PBDB on different groups. Long story short, this is what we found. We found that the rates of extinction for the marine megafauna in the Pliocene were much higher, significantly higher than the mean Cenozoic and any other epoch. And the Pleistocene was also quite high. And when we looked at this at continuous time frame or a continuous time series, we found that it was actually the late Pliocene when extinction rates were the highest. When we looked at proportional extinction of the marine megafauna of the time, we found that, again, extinction is not random. Some species, some clades were affected more than others. Mammals lost around 50% of the genera, but sharks only 10%. And then you have seabirds and turtles, but overall, we lost 1/3 of the genera in this extinction in the Pliocene. So that's the number of the proportion of genera that didn't cross from didn't make it from the Pliocene to the Pleistocene. And most of the species were coastal, not because extinctions were most concentrated in the coastal environments, but because most of the species are actually coastal. They live in coastal environments, which are the most productive one, and support it's an environment that can support the feeding needs of these large species. But again, the question that we were after is beyond numbers. What happens when we lose 1/3 of the marine megafauna in the Pliocene? So we use this approach, the functional diversity approach, that ever since I've been using nonstop for multiple studies, and I'm going to show some other results on this for modern species. But basically, is based on the measurement or on the assignment of functional traits, being those traits that determine what do you do in ecosystems, how do you use resources. So diet, habitat, body size, and so on. And then we use a principal coordinate analysis to create a functional space. And then the volume of the space and the distribution of species or functions within the space can allow us to measure different indices of functional diversity. So we use this approach for the Pliocene and the Pleistocene, calculate the functional richness, the volume of the space, and functional redundancy, the number of species per function. And this is what we found. So we found that we lost 1/3 of the genera, but this translated into a loss of 15% of the extent of this functional space, of the extent of functions, and a 20% shift. And I'm showing here these two spaces. And of course, that you can really tell the 15% difference by looking at these plots, but I'm showing just 2 dimensions, and the dimensions of this functional space was 6-dimensional. And also we did a lot of iterations to account for sampling differences. But long story short, we found that the loss of 1/3 of the diversity resulted in the loss of 15% of the extent of ecological functions, and also 7, is it 7 or 8? I think 7 functions, like unique trait combinations, let's call that functions. So say, large, benthic, so on and so forth, were also lost. And so this, it wasn't a mass extinction event, of course, but these numbers are really critical. They're very big when compared with other studies that had used similar approaches. So I'm not going to talk much about it, but other studies have shown that very little changes in functional diversity, even after a mass extinction. And here we have a 15%, 7 functional entities, so it's quite significant. So we propose that this kind of disproportionate loss of functions of ecologies was due to the low functional redundancy of this marine megafauna. So here in these histograms, I'm showing how many clades you have per unique trait combination. Again, let's call it functions. And there was an average 2 or 3 species, or 1 or 2 species, no, 3 species, sorry, I have it there in my little drawing, per function. So of course, when you lose one species, chances are that you're going to lose the whole function. And to compare and to assess if the role of functional redundancy in determining the overall effects after extinction, we did a similar study with the mollusks from Panama. We call this project Pirates of the Caribbean, because we studied mollusks of the Caribbean using Pirate, which is the software. And this is a well-studied kind of extinction event that took place in these benthic communities. And we saw that, you can see here in this plot F, that despite a very important extinction of 50% of the species of mollusks alone, there were almost no changes in functional diversity in the extent of the functional range. And of course, the redundancy was quite different. So to put those 2 studies together, we found that in the marine megafauna, when we had really low redundancy, just a few species playing a role, a lot of unique roles playing just by one species, versus the mollusks, where we had an average of 7 species per function, resulted in much lower loss of functional diversity compared to the taxonomic loss. So the taxonomic loss of marine megafauna was 1/3 of the diversity, and in terms of mollusks was 50%. Yet in terms of the mollusk, for the mollusk, we lost a tiny part of it, 3%, whereas for the megafauna, we lost 15%. So functional redundancy is it's a key factor that determines the ecological consequences in marine ecosystems. And of course, the more redundancy you have, the most buffering capacity you have as well in the face of extinction. But one of the main questions, of course, that remained from the study of the marine megafauna is what caused this extinction. And because of the work that we had done with the megalodon, we knew that the temperature didn't really directly affect these kinds of animals. And I have to say, this was Carlos Jaramillo's idea, that we needed to look into area, because he had the suspicion that area had something very important to do with this, and he was right. So to go to area, to assess the effect of area, changes in area in these extinctions, we looked at sea level changes, and we used these 2 approaches, these 2 curves. And in collaboration with with Sara Varela, my friend from Berlin, we took the last 5 million years. And what Sara did was to kind of grid the whole world for all of these scenarios and assess the amount of neritic area of this coastal habitats, over 200 meters, that was left in the aftermath of these increases and decreases in sea level changes that have been well studied. You can see this curve was from 2005. And what we found, this is a graph for area, is that the area available, the coastal habitats available, areas over 200 meters of depth, from the Pliocene to the Pleistocene, dramatically declined in the late Pliocene. And not only that, but they were very, very variant throughout the Pleistocene. And in average, it was 30%, it was 3 times more variable. It was 30% less in the Pleistocene than the Pliocene, the area available, the coastal habitats available. And there were times where it was even 50% less. And we did some analysis where we wanted to know which were the traits that better predicted these extinctions. And we found that it wasn't body size or habitat or diet, but it was thermoregulation, with homeothermic animals having higher chances of going extinct. And we think this corroborated the hypothesis of area loss having an effect on this extinction, because homeothermic animals, because they need to regulate their body temperatures, and this is the marine megafauna, so they were large, they needed to eat a lot more than ectothermic animals. And they needed good quality prey, like a lot of, like in the case of a megalodon, a lot of fatty animals. And the less area you have, less chances you can find this kind of prey. So in summary, we found that the loss of habitat likely determined or was a key factor in the extinction of marine megafauna, affecting the most homeothermic animals. And of course, that the functional redundancy was a key factor in determining the ecological consequences of this extinction. But I just want to emphasize on this part here, because to me, at the time when we found that homeothermic animals had more chances of going extinct, was quite puzzling, because there was this other study that had been published around the same time, showing that homeothermy was a key factor in driving the evolution of giantism in the megalodon. So we did this other analysis, which I'm going to talk about just briefly, where we mapped the evolution of homeothermy and filter feeding, the 2 types of feeding mechanisms in elasmobranchs. We found that filter feeders and homeothermic animals had evolved significantly larger sizes than the rest of sharks, the ones that were normal, ectothermic, and macropredators. We also found that mesothermy evolved once a really long time ago, 140 million years ago, and filter feeding around 4 times, about 4 times around 50 million years ago. And this corroborates previous studies. This wasn't quite of a surprise. But the appearance of mesothermy occurred in a time of global cooling, where being able to regulate, by the way, mesothermy means being able to regulate the body temperature of the most important organs, and that allows you to live in colder water. So of course, in a time of global cooling, this allows you to expand your niche, and also allows, because the muscles get warm faster and you can have faster burst speeds. So enhance prey intake in terms of cooling. And in terms of filter feeding, evolving 4 times, it was in times of increased productivity, when plankton was available, it was abundant. So both of these adaptations that allowed, or were kind of tied up with being a giant, occurred when there was enough quality and abundance of prey, and allow the species to enhance prey intake. And I just wanted to bring up that we lost the megalodon because area wasn't enough. The area available wasn't enough for the megalodon to find enough prey, but also because probably a lot of its prey went extinct. I don't know what was the order of events, but the prey declined. Area was not available as much, having all these metabolic demands. New competitors came, increasing the pressures, and this giant is lost forever. And we have today still the other giant, the whale shark, who is a filter feeder. And of course, is being threatened today by this microplastics problem that we have. And the very fact that this species is a filter feeder it was a key factor that allowed it to become giant, and now is the one factor that is threatening the species, is the fact that it eats filter feeds. So yeah, this is just to transition to the 6th mass extinction that I talked about at the beginning. And when we did this marine megafauna work, we were wondering, so we used to think, when we were looking, when I was writing the introduction of this paper, I realized that we had this perception of the marine megafauna that we have today being pristine. We came to this world as humans and we inherited this pristine marine megafauna, but in fact, it had gone through an extinction event in the recent geological time, right? We received an already depleted marine megafauna. So what is going to happen to this fauna in the future when we're going through this overfishing and habitat loss, climate change, and so on, the fate and function of the marine megafauna? Oh, I should have put this before. So yeah, the marine megafauna is highly at risk, especially sharks. Like I said at the beginning, is one of the most threatened marine vertebrates groups that we have today. So to answer this question, once again, we use the functional diversity approach, in this case with extant species of marine megafauna, which are 134 species of over 45 kilograms. We assign 10 functional traits, and we build the functional space to assess functional richness, but in this case, using a slightly different approach, all the functional diversity metrics were based on this space. So we assessed functional uniqueness, then the distance of species to its closest neighbors, which tell us how redundant they are, and functional specialization, how close to the centroid of the space they are, which tell us how extreme they are and how important they are for maintaining this volume that is so important. And we also use the IUCN status of each species to simulate some extinction scenarios. Some of them associated with time, in this case, 100 years. So each extinction status in the IUCN has a probability of extinction through time. For example, a number of species going extinct in 100 years. And we also use another scenario that was very pessimistic yet realistic, and is that what happens if the status quo of conservation is maintained, all species that are currently threatened go extinct. And this is what we found. So for the scenario in which we use the probabilities of extinction in 100 years, we found this, but I'm not going to talk about it because let's focus on the most dramatic scenario for the sake of time. So for the scenario in which we assume that all species that are currently threatened will go extinct, we found that we may be losing around 40% of the marine megafauna, and this translates into almost 50% of the extent of the functional space, and an increase of 15% of the functional uniqueness, so less and less redundant. And what is showing here with the violin plots is that all of these results are much higher, much different than expected by chance. So in terms of the number of species that we'll be losing is very similar to what we found, or higher actually, than when we found in the Pliocene. So 1/3 of the megafauna going extinct, and also much higher than the 50% change that we found from the Pliocene to the Pleistocene. In this case, we're talking about 50% difference. So yeah, this is looking really bad for the marine megafauna, especially for sharks. And I was going to talk about sharks in particular, but I won't because of the time. But I just say that sharks have the highest projected losses of functional diversity, 80% or something like that. But so we proposed a new conservation metric that was inspired by the EDGE metric that is based on on the functional contributions of species. This is aiming to not only preserve species based on whether or not they are threatened, but how important they are to maintain the functional diversity of this fauna. So one of these components for this metric is functional. The metric's name is, and the problem is I can see some of my slides because of the Zoom window, but okay, I can see it now. I just moved it. It's called FUSE, which means functional, unique, specialized, and endangered. So functional unique, why we incorporate the functional uniqueness is because, like I said, is the distance to these 5 closest neighbors. So for example, here we have 2 examples, one species that is orange and one that is yellow. And if you lose one species, the one that is orange, and then you will be losing this part of the functional space, you leave that gap. But if you lose this one that has very distant neighbors and therefore is not a very redundant one, then you leave a really big gap in this functional space. So we want to avoid these big gaps. So the loss of functional, because the loss of functional unique species results in big gaps, right? And in terms of specialization, the distance to the centroid, if we lose a species that is very close to the centroid, and then you measure the extent of the space remains the same. But if you lose a species that is very close to the edges, and you try to measure the space again, the extent of the volume, then you lose a lot of space. So we also want to avoid the loss of specialized species because that results in a loss of volume. So we combine these two metrics with the IUCN status, similar to the H metric that is based on evolutionary distinctiveness. We highlighted the most functionally unique, specialized and species, marine megafauna. And for example, here we show the top 5, the green sea otter, Julian's golden carp, the dugong, the sea otter, and the giant clam as being the marine megafaunal species that contribute the most to functional diversity or disproportionately contribute to functional diversity, but also are threatened. So that's for FUSE. And just very quickly, Ariana, our master student here at Swansea, is now mapping this in the whole kind of biogeographical context. See where are the hotspots for functional diversity for the marine megafauna, but also where few species distribute. And as you can see here, there's a lot of important species in pelagic zones and in the high seas, which is quite interesting for marine megafauna. So stay tuned for Ariana's amazing work. And I already did this. So this other project that I'm working on at the moment, we call the Future, because a lot of these projects ask you to come up with an acronym, and this is not a great one, but Future sounds cool, which is the fate and function of threatened fauna and flora in the Anthropocene. And the whole point of this project is to apply FUSE to other taxa. So we're doing it with palms. So I'm going to the botanical world, marine mammals, and of course, sharks. And I am going to show you our results for sharks. So we built a functional space for all the extant species of sharks. And here's the functional space, for example, of sharks versus rays and skates, showing that they have a very similar extent of volume, but also for each order of elasmobranchs and these lamniforms is particularly important, but I don't think I have time to talk about it. But here, we show how the functional space of threatened species is basically the 100% of the total extent of the functional space, whereas no threatened species hold only like 36% of the space. So that's quite important. And here, this is that dozens, the top 12 few species. So you have the Chilean ray as being like one of the highest priority for the preservation of sharks in terms of functional diversity. But what I wanted to highlight here is there's only one species that we share with the top H species, showing that this FUSE facet of biodiversity is complementary to other metrics that we're using to protect species. So you don't have the same set of species because here we're trying to protect a different facet of biodiversity. We also plotted the hotspots for functional richness. And well, not surprising to have like the Caribbean being a hotspot for functional richness for sharks, but very interestingly, a lot of oceanic islands that had never come up as hotspots for the protection of sharks come up as such when you focus on functional diversity. So this is where functional diversity concentrates the most, the top 2.5% of values. And here just to show that when you compare it with other hotspots of species richness or phylogenetic diversity, there's a lot of unique hotspots that are only for functional diversity, and because this is an understudy facet of biodiversity. And this is where top FUSE and H species are. And in orange are the congruous zone, and in red are the ones that are only for FUSE. And again, the Caribbean, specifically the coast of Colombia and Venezuela, come up as important where we have the most FUSE species, but also unique in terms of this not a hotspot for other kinds of species. So this is something I'm currently working on, really hoping to submit very soon. And this is also kind of overlapping the hotspots for FUSE and H with fishing. And as you can see here, the Sea of China comes up as a very important part of the world where you have a lot of uniquely functional, unique, specialized, and endangered, and evolutionary distinct and fisheries. So this should be an area of focus to protect these important species. And just to end, I wanted to show some results on the new project that I'm doing in Zurich, which is called SharksXT, Sharks Extinction Mechanisms. I was lucky enough to get this grant and then be able to hire this amazing group of people. And they are already producing amazing results, but I actually wouldn't make it justice if I would try to present them here, because they are really interesting, but also very sophisticated. And I think in the future, if you can hear from them directly, it will be much better. And yeah, so basically, the point of this new project, Shark Extinction Mechanisms, and in general of the research that I've been conducting at the moment, is so that we can have a more comprehensive and complete view of extinctions, shifting the focus a bit from benthic to vertebrates, as well as covering like global extent, and also go really deep in time. So as you saw, one of the first slides that I show, we've been gathering information for all the Neoselacians from the Cretaceous to today, and stay tuned for the results of that really cool research that Cristina, Amanda, Gregor, and Jaime are working on. And I think I should end now, and I just want to thank, obviously, my group, which includes also the group of Daniela Silvestro. We collaborate very closely, and Rachel Warnock as well. This is one of our joint lab retreats that we had last year in Switzerland. And also my collaborators on the different projects that I talked about today. And I think that with that, I'll have a cup of tea because my mouth is very dry, and take any questions. Thank you very much.
Thanks, Catalina. That's perfect timing. It's 10 minutes too. So you take a cup of tea while we'll set up for the questions. So please, thanks, Catalina. That was a great talk. We have one question already coming in, and I'll let Paul say that question himself. But if you have a question, please either write it in the Q&A or raise your hand using the button below. While Paul, I move you to ask your question yourself. Paul, are you there? Can you hear us? Can you speak? This is Paul Harnick. He says, I'm not sure I have audio. Okay, I'll ask the question for you. Okay, so Catalina, he says, this is Paul Harnick. Catalina, great talk. In your study comparing functional shifts between megafauna and mollusks in the Caribbean, did you also compare extinction rates for these groups? And if so, which group was most sensitive to changes in habitat and or productivity? I think you can also read that question too.
Yeah, that's a great question. We didn't look at it under that lens. So the marine megafauna was like a separate study, and the mollusk was another one. And we looked into extinction rates for mollusks, but they had a different taxonomic resolution species, and we did it by genera for the megafauna, but didn't really compare the magnitude of these extinctions. And I don't, yeah, if I had the two plots, I could look at them and tell you, but yeah, I don't remember. And for the marine megafauna, we didn't really look at the potential environmental drivers of extinction, but for the mollusks, we did look at other environmental factors, and we found that, if I remember correctly, it was, I think it was sea level as well, one correlated the most, but actually it was like a secondary result, and that's why it's not in the top of my head at the moment, because what came up as the most important predictor of extinction was actually redundancy, with species that were highly redundant going extinct the most, which explains also why we had such a low level of functional diversity richness. But yeah, to answer the question, we didn't really look, and we didn't really compare the two studies like that. I'm doing it for the talk, but they were two separate studies, really. But it would be really great if we can do that.
Thanks, Catalina. So Paul says, thanks for your answer. Any more questions for Catalina? The floor is open. Does the banana produce bananas?
I wish, no. No, I still have to buy bananas from Colombia and Panama.
Nick says, I always learn so much listening to the fantastic work that you and your group do. This has been a great overview and a dense download.
Thanks, Nick. I'm so glad that Nick managed to stay until the end. And thanks, Paul, as well for your questions.
Any more questions for Catalina before we finalize today's seminar? Ian says, thanks for the talk. Can't wait to read more and illustrate some of these ideas. Great talk. Lots of nice comments coming in. Yeah, it's great stuff, Catalina. As always.
Thank you, everybody. Thank you, everybody, for coming.
Yeah, thanks everyone for joining us. Especially thanks to Catalina for giving us this Q-Mare seminar. So next month, we have another wonderful speaker, Edin Sau, who's going to be talking about the emergence of latitudinal biodiversity gradients in deep time. Another really important question is, aside from extinction, changes in biogeographic patterns associated with climate change. I think she'll be dealing with that. So that's something that the fossil record can help us with, I think. So don't miss that. That's on March the 1st. With that, I'd just like to thank Catalina again for your wonderful talk and for everyone for joining. Have a great day.